Hi, I'm Bjoern Sill from the laboratory of Dr. Cowan in the Department of Anesthesiology at Children's Hospital and Harvard Medical School. Today we will show a procedure of optical mapping in Langendorff-Perfused red hearts. We use this procedure in our laboratory to study the spread of electrical activity on the cardiac surface. So let's get started.
On the morning of the experiment, mix up 4 liters of Krebs Henselite solution. After mixing up the solution, remove undissolved particles with a 200 micron filter using vacuum filtration. First, use 1 liter of the prepared Krebs Henselite solution to dissolve 11 millimoles of BDM. Second, take another 100 milliliters of Krebs Henselite and mix in 10 millimolar di-8-aneps in DMSO to a final concentration of 5 micromolar di-8-aneps in solution. The water-jacketed glass reservoirs, the bubbler, and the glassware used here are all from Red Naughty. Transfer the 2 solutions to the reservoirs pre-warmed to 41 degrees Celsius. Oxygenate the solutions with 0.2 micron filtered gas using a submerged bubbler. Pump the solutions from the 3 stock reservoirs through the wall-mounted Langendorff system with peristaltic pumps and low-absorption silicone tubing.